Hey there, biology. So today we're going to be taking a look at a process called protein synthesis. Protein synthesis is the process by which our cells use our genetic information, our DNA, to build the proteins they need to carry out the necessary functions of life. You may remember from earlier in the year that proteins were one of the 4 macromolecules. Proteins are important in virtually everything that cells do. So if you think about those 8 characteristics of life that we learned about, like responding to stimuli and maintaining homeostasis and growing and developing and reproducing and utilizing energy and so on, proteins are crucial to all of those characteristics of life. Now, if you want to read more about protein synthesis, you can take a look at it in section 14.2 in your textbook. Before we dive into protein synthesis, though, I just want to do a quick recap on the differences between the 2 different kinds of nucleic acids. There are DNA and RNA. Now, they're really similar in a lot of ways, but there are 3 key differences between them. So between DNA and RNA, both of those molecules have a sugar-phosphate backbone, alternating sugars and phosphates. But the difference between them is the sugar molecule itself. DNA contains a sugar called deoxyribose. RNA contains a sugar called ribose. The phosphates are the same. It's the sugars that are different in the backbone. Now, the bases are also different in DNA versus RNA, at least in one way. Guanine, cytosine, and adenine are found in both DNA and RNA, but thymine is only found in DNA, and in RNA, instead, we would find a base called uracil. The final difference between DNA and RNA is that DNA consists of 2 strands of macromolecule twisted around itself. That's the shape that we call a double helix. RNA, on the other hand, is just a single-stranded molecule. All right, so now that we've reviewed the differences between DNA and RNA, let's go ahead and dive into the basic idea behind protein synthesis. And that is that DNA is used to make RNA, and then in a second step, RNA is used to make proteins. That means that the process of protein synthesis is actually a 2-step process. That 2-step process is illustrated here, where we see DNA with the blue arrow leading to RNA, and then with the green arrow leading to a protein. That blue arrow represents the first step in the process called transcription. The green arrow represents a second step in the process called translation. So what we're now going to do is we're going to take a look at the transcription process first by going through the steps that are involved. Then we'll watch a short animation on how transcription works. So let's move right into that. Transcription, first of all, takes place in the cell's nucleus. The reason why that is because that's where the DNA is found. So transcription occurs in the nucleus with the help of an enzyme called RNA polymerase. RNA polymerase is an enzyme that manufactures molecules of RNA. And it does this by using information from DNA as a template. And it matches up the base pairs, creating a molecule called messenger RNA. Now, once that messenger RNA molecule is created, it can then leave the nucleus and go on to the second step called translation. So here's an illustration that shows us the process of transcription. Within the nucleus here, we have our DNA molecule. Now, it's important to note here that we are looking at a tiny, tiny, tiny fraction of the amount of DNA that would be used in transcription. A real gene would be many, many hundreds or maybe even thousands of nucleotides long. So the fact that we're only looking at 9 nucleotides here means that we're looking at just a small representation of the entire process. The transcription step involves base pairing nucleotides as we normally would, except we're using uracils instead of thymines. So in DNA, we have a thymine, which would base pair with an adenine in RNA. In DNA, we have an adenine, which would have paired with a thymine, but because there's no such thing as thymine in RNA, we get a uracil instead. Guanine pairs with cytosine, adenine pairs with uracil, cytosine pairs with guanine, and so on. So this is the first step in creating a protein. We're getting the information that's stored in DNA as a copy of a molecule called messenger RNA. So now we're going to go ahead and watch a short, simple animation that shows us how this process actually works.
When transcription starts, a small section of DNA is unwound. One of the two unwound strands acts as a template for making the message. The messenger RNA is built up, one nucleotide at a time, according to the familiar base pairing rules. A on the DNA pairs with U on the RNA, G pairs with C, T pairs with A, and so on. The result? A message with a base sequence complementary to the template strand of the DNA. This messenger RNA will eventually be used to direct the formation of a protein.
So now that transcription is complete and the messenger RNA has exited the nucleus, we can take a look at how translation works. But before I do so, I just want to say that translation is a little bit more complex than transcription. So like before, I'm going to go through the steps of translation here in the PowerPoint, then we'll take a look at an animation, and I think once you get both of those two things together, the description and the animation, it's going to make the process really clear to you. All right, so we're out in the cytoplasm, and we're going to use a ribosome. You may remember from earlier in the year when we were learning about cell parts that ribosomes are the sites of protein synthesis. So now we're going to see exactly what the ribosome does. Now, the ribosome has a bunch of different jobs to carry out in the translation process, but its first job is to identify a special place in the messenger RNA where translation should actually begin. That's called a start codon. The start codon is a 3-nucleotide sequence of bases in messenger RNA that identifies where the ribosome should begin the translation process. Once the ribosome has identified the start codon, then the first transfer RNA will arrive. Now, the T in tRNA stands for transfer, and what it's transferring are amino acids. It's the job of transfer RNAs to bring amino acids to the ribosome so the ribosome can put them together and assemble a protein. Now, the way that the transfer RNAs and the messenger RNAs connect to one another is because messenger RNAs have codons and transfer RNAs have anticodons. They're both 3 nucleotides long and they're complementary to one another. So a codon on a messenger RNA is complementary to an anticodon on a transfer RNA. After that first transfer RNA arrives, the second transfer RNA with the appropriate anticodon arrives as well. The ribosome is then going to take those amino acids that the transfer RNAs are bringing and join them together. Now, this process is going to continue over and over and over again, the ribosome taking amino acids from transfer RNAs and joining them together until eventually a special kind of codon is encountered called the stop codon. The stop codon identifies to the ribosome that it should stop the process of translation because the chain of amino acids is now complete. Here's a good way of thinking about how start codons and stop codons work. Let's say you have a teacher assign you a book to read, but they only want you to read a portion of that book. So your teacher tells you, I want you to read pages 10 through 20. Start on page 10 and stop on page 20. What does that mean you're going to do with pages 1 through 9? Nothing, because you're not going to start until you have the instructions to start, which is on page 10. What about pages after page 20, like 21 and 22? What do you do with those? Nothing, because your instructions were to stop on page 20. So the ribosome, just like you reading the book, will start when it encounters a start codon and will stop when it encounters a stop codon. So here's a diagram from your textbook that illustrates this. We're going to watch the video on it in just a second, but I want you to notice here that this sequence right here called AUG, that is a codon. This is a special codon. This is actually the start codon. Here's another codon, UUC. Here's another codon, adenine, adenine, adenine. And these molecules up here, these are the transfer RNA molecules. They're carrying with them the amino acids that, as you can see, are in the process of being joined together. Now, a real protein is many, many dozens, hundreds, or in some cases even thousands of amino acids long. So what we're observing here is just a small representation of what actually happens in protein synthesis. So let's go ahead and take a look at that animation now.
It turns out that each triplet of bases on the messenger RNA, called a codon, corresponds to a particular amino acid. Now there has to be a chemical connection between each triplet codon and each amino acid. A special adapter molecule called transfer RNA makes that connection. One end of the adapter carries a particular triplet of bases called an anticodon. This matches up with a specific triplet codon on the messenger RNA. The other end of the transfer RNA is capable of binding to the unique amino acid corresponding to that anticodon. Since there are 20 amino acids, there must be at least 20 different transfer RNA adapters. So, for a protein chain to be assembled, each triplet codon on the message is read. The transfer RNA adapter with the relevant anticodon binds the messenger. The amino acids at the other end of the transfer RNA adapters become joined by a peptide bond. Once a transfer RNA molecule is no longer needed, it's released. The process repeats, elongating the peptide chain until a stop codon is reached.
Okay, so that animation does a really great job of showing a simplified process of translation. How the transfer RNAs are compatible with the messenger RNA, because the transfer RNAs have anticodons that are complementary to the messenger RNA's codons, and how those transfer RNAs are carrying amino acids to the ribosome so that they can be linked together in a long chain of amino acids. Now, let's go ahead and take a look at a summary of this entire process. Here I have some DNA, a small region of a gene, and I know this is DNA for a couple of reasons. One is because there are two strands, and DNA has two strands. And the other reason is because there's thymine in it. And remember, RNA doesn't have thymine, only DNA does. So the two different strands of DNA that we see here, one of them is called the non-template strand, and that's the one that's not going to be copied. So what I'm going to do is I'm just going to hide it. It's still there. I just want to declutter the screen here. So this is the strand that's going to be used as a template to create messenger RNA. So this process here, where nucleotides are being paired up, is called transcription, and that's being carried out, of course, by RNA polymerase. Once this messenger RNA is constructed, this molecule would then leave the nucleus and head out into the cytoplasm, where the ribosome is going to attempt to identify the start codon, and there it is. AUG is the start codon, and once that start codon is encountered, then the messenger RNA will be translated exactly 3 nucleotides at a time, 1 codon. Now that the start codon has been identified, the first transfer RNA is going to arrive, and it has a complementary anticodon. If a codon is AUG, then UAC has to be the anticodon. That means that the next codon is going to be UCU, then the next one is UCA, then AGG, then CCC, then UUG, UCG, and then UAG happens to be a stop codon. So what we're observing here is a small representation of real protein synthesis, because to produce a real protein would take many more amino acids than the 7 that are listed right there. Now, before we wrap this up, I just want to point something out that some of you may be looking at down at the bottom of the screen there and wondering, wait a minute, polypeptide, protein, what the heck is the difference? Well, they're really similar to each other, but they're not exactly the same. A polypeptide is simply a bunch of amino acids linked together. You may remember from that animation that we just watched that the bond between amino acids is called a peptide bond. So if I have many peptide bonds, because there are many amino acids linked together, that's what a polypeptide is. Now, once translation is completely done, that polypeptide will begin to fold and twist and bend and connect to itself and take on a three-dimensional structure. That three-dimensional structure, that's what a protein is. So they're really similar, but they're not exactly the same. Now, the last thing we're going to take a look at is how it is that these codons up here can correspond to a particular amino acid. How is it that adenine, guanine, guanine corresponds to arginine, or cytosine, cytosine, cytosine corresponds to proline? That's where this comes in. This is called a codon chart. Now, this codon chart was put together by scientists after researching how translation works over a number of years, and they were able to figure out exactly what three-letter sequences of messenger RNA, those codons, correspond to which particular amino acid. So here's how this codon chart works. We're going to start at the center of this chart, and we're going to take a look at a few examples. So let's say, for example, I have a codon that is a sequence CAU. Here's how this codon chart works. We start in the center, and I'm going to choose C because that's our first base in the codon. Then I'm going to move out to the A because that's our second base, and then I go to the U. So that means that histidine is going to be the amino acid that corresponds to this codon. Let's take a look at another example. Starting in the center of the wheel again, GAC. We go to the G, we move out to the A, and then we move out to the C. That means that GAC corresponds to aspartic acid. Let's take a look at one final example. This time it's UAG. So we're going to start in the center, we're going to move to the U, then we're going to move to the A right here, and then we go to the G, and that one is one of the three special stop codons. UAA, UAG, and UGA. Those are stop codons that are special as compared to the rest of these. Okay? Now, there's another way that codon charts are sometimes illustrated, and this is with the rectangular one. I prefer the circular one. It's the one your textbook has, but if you were to look up codon charts on Google, you may find this one. But it works exactly the same way. Down the left-hand side here, we have the first base of the codon, then across the top is the second base, then down the right-hand side is the third base. So if I had, for example, UAC, I'm going to take a look at the U section, so we're somewhere in this very top row. And then my second base is A, so I'm going to be somewhere in this box, and my third base is C, so that means it's got to be tyrosine. Okay? If we were to look up, for example, CGC, we're going to look in the second row now. Then we're going to look at this last column, and then the C tells us that it's got to be arginine. CGC is arginine. Now the last one is GUA, our last example here. So G, and U is the first column. GUA is valine. Now, I don't want you to worry about memorizing the codon charts, the square or the circle, but you do need to know how to read these. So make sure that you understand how to read either the circular chart or the rectangular chart. Okay? All right, that pretty much does it for protein synthesis. So until next time, take care.